Welcome back to studying the last topic of chapter 2, microscopy and specimen preparation. So the newer techniques in studying microscopy or in doing microscopy, exactly two types of microscopy techniques we are going to study today, and that is the confocal microscopy and the other one is scanning probe microscopy. While in scanning probe microscopy we have further further types of atomic force microscope, further two types of microscopes. So I'll just give you a general idea. Usually I read it completely, but today I'm not going to. I think this is a new method and so people don't get bored. So when we talk about newer techniques in microscopy, we are talking about the techniques that are not used that much commonly, or you can say like most people don't have. It is an advanced technique and it's not your general microscope, right? The construction is a bit different than the normal microscope. It has the addition of one thing or two things, or the addition of a completely new concept in them. So in confocal microscopy, they are saying that by comparison with the light microscope, when you see an object in a light microscope, right, the light comes on it, right? Just think about, I know this is scanning electron microscope, but think about this. The light goes on a specimen and it scatters in all directions in the light microscope, and when this scattered light is taken up by the objective lens and when it reaches your eyes, so the object looks blurry and not that much clear, and some objects even look kind of fake. It's a 3D diagram usually. But the confocal microscopy actually takes your picture, it removes the scattered line, well, it will be shown here. It removes the scattered area and it only takes the light from one focal length, and then it removes the extra line, and then there is another lens other than the objective lens. Upper to the objective lens there is another lens, and it only leaves the light that is only coming from one plane of focus, and that's why your image becomes more clear. Now let me tell you about the other one as well. We will explain it afterwards, right? Now the scanning probe microscope. So its concept is scanning. By scanning you can guess it's scanning just the surface of the molecule, but it is on atomic level, right? So you have a kind of probe and it has a tip, and the tip has atoms on it and it generates electricity. That tip, it tunnels the electricity, right? Well, in one kind of tunneling microscope, it tunnels the electricity and it is so sensitive that it can see up till the atoms of the surface as well, right? Usually we just see the structures and we see it from the reflected rays or the refracted rays, but in case of scanning probe microscopy, we see the atomic structure of the surface. Like look at this diagram, it has DNA. The DNA, you guys know that it has nitrogenous sugar, phosphate, hydrogen bonding. Even the hydrogen bonding is clear in that, and it does by putting the electrons in them or by putting pressure in them. Like there are two types of them. So this was the general idea what we are going to do, right? So now let me go back and tell you step by step what actually is happening in them. So confocal microscopy, it actually starts from here. What am I doing? Biological specimens are three-dimensional. This is the basic concept in the confocal. Like our objects are not two-dimensional, not one-dimensional, they are three-dimensional. Means they scatter light from three-dimensional, from X, Y, and Z. So usually in light microscope, they see objects from all angles together at one point, right? And that's why there are multiple kinds of unwanted information in that, and we don't want that because it blurs the originality of the picture and we can see clearly. But in confocal microscope, as you can see in the first diagram, it sees from three different planes. It only studies one plane at a time. Like it can only see X plane, Y plane, or you can say the Z plane. The specimen is in 3D structure, right? Here, let's let me explain this diagram. So consider this is your specimen, right? Consider this is your specimen, this is the cell, right? And you want to study the cell DNA, right? Now when you put this light, this is the source of light, just the same way as your light microscope has a source. So you have laser light and it is going towards your cell. Originally there are reflected rays from it, right? Reflected light. Now there are two types of reflected light that are coming out of it. The one is the rays or the light that comes from the plane of focus, only one plane, either X or either Y or either Z. The other things up the plane and below the plane are not considered the rays that we wanted. So if the rays that come directly to the plane of focus and reflects from that, we consider that okay, we want that light. But other than that, the lights that come from the upper area or the lower area of the plane of focus, we want to delete them. So when it goes pass through the objective lens, there is another lens here, right? Like there is a scanner, it scans which light are the red lines are actually the lines that we want, and it comes only from one plane of focus, right? Plane of focus means the only plane that we want to consider, right? This scanner know what rays are coming from the plane of focus and the blue ones. So when it passes through the other lens, it kind of separates them. Like here it's not that differentiated, but here it kind of, you see, it changed the angle of it somehow, and there is the aperture, and so the lens removed the trajectory of it, right? Like change the trajectory of the blue lines, and it cannot pass through this small hole that will lead to your detector. Or you can say usually in confocal the computer detects us, not our eyes, because it is a laser light, so it stops the blue rays while it allows the red ones to go further. So it kind of works like that. Now this is confocal scanning laser microscope. Why is it laser microscope? Because the source of light that is used is this scanning laser. Now look at this. There are in this diagram they are explaining to us what's happening. Now if you take two beads, right, normal two beads, and you see it under the light microscopy, look, it shows the one image, well, kind of, blurry, right? Like you can see the surface, it's kind of the same the whole time, and it's not the same. Well, look at the other bead. You can't even consider if it is a bead or not. You might think that this is just a conjectured source of light or something. So in light microscopy, this is how we see the beads, while in this one, wait, what was I saying? So while in this one we can see the light from all angles, and you can see that these two are the beads. By the way, the CSLM is confocal scanning laser microscope. So this is not the 3D construction, this is just how it is reflected from plane by plane, right? Well, this is the 3D construction. Like this is how you see it in the confocal microscope, and this is how your computer constructs it, right? Because the detector is connected with the computer. So this is how it constructs its 3D. Well, look at this one. These are showing the sections of it, because we studied that in the confocal scanning laser microscope we do study the plane separately. So this is how the Z cross section looks like cut it just as this way, cross this line, you can say this angle is your Z axis. So this is how you are seeing them, right? I don't know if it makes sense, but it makes sense. Now you can use this confocal scanning laser microscope in studying the biofilms. Guys, biofilms are the layers of the same bacterial colony. They definitely are not connected to one another, but they are so close to one another and they signal one another. That's why biofilms are more resistant in causing problems and are so difficult to get rid of. Now, if you see, they did use the fluorescent. Give me a second. There is a noise coming. I need to get rid of that. So yeah, the noise was way too loud. So in case of biofilms, right, 3D reconstruction of biofilm, so they did use the fluorescence or the stain, right? So the red ones are the dead ones, and the green ones are the ones that is alive. So you can see, this is the picture of a medical equipment, and this is, you can see, a biofilm is made from it. So this kind of microscope is used in the medical operator seeing, because we want to get rid of these biofilms. It is important. So if you want to read it, you can read it. It's all the same thing, right? The biofilm was exposed to antimicrobial and then stained with the dyes that distinguish living from the dead cell. Yeah. So it looks like they did expose it with the antimicrobial agents, but like I told you, that biofilms are so resistant that even if you put the antimicrobial agents on them, they survive. So look how many of them survived. Now, looks like this is it with a confocal lasing microscope. So it was not that difficult, but it just shows us that there is a better microscope than the light microscope for us to use, with only one lens and one scanner more, and a computer needs to be attached to it to show us more clear object than the light microscope, right? And this was actually what the scientist is shown or told, or they told us when was it developed, right? No, they didn't tell us when, but I think it's not that, it's not in this age. It has quite some time, because this scanning probe microscope is way more advanced and it works on the atomic level, but it was developed in 1986. Can you believe it? 1986, guys. This is 2025. So although the light and electron microscope have become quite sophisticated and have reached an advanced state of development, we know that electron microscope can see quite deeply into an object to organelles level, right? But we need to go to the atomic level. How did we discover atoms? How? Guys, you need to think, how did the atoms come out of nowhere? I know there are theories that there is something that shows that there are atoms, but can we see it? Yes. Because of the scanning probe microscopy, we can see it. So if the laser microscope were making images more clearer, this one is reaching to the depths of that surface to the atomic level. Now there are two types: scanning tunneling microscope and the atomic force microscope. Both shows the atomic level, but their mechanism is just a little bit different. Now they are saying that the scanning tunneling microscope has the magnification of 100 million. Guys, 100 million is no joke. I think 100,000 times is the ability of the electron microscope. So this is way more than that. So the electron surrounding, why am I explaining or reading? So in this kind of microscope here, let me explain it from this. Even though this is atomic force one, right? So there is this cantilever and it has a tip attached to it, right? The tip has the atoms on it, and this is, as you can see, the slide, it has specimen on it, and you want to see the DNA, right? You put the laser, in this case this is the laser, but in this case the tunneling microscope, it has electricity attached to it, and when the electricity goes from that tip and it tunnels into the specimen, and it reaches to the atomic levels. When it reaches to the surface of the atomic level, it sends the signals into the cells and separates them. Right. The tunneling signals is so sensitive that even if you take it a little bit further, like a half diameter of the cell, it will stop working and it will show the image of another area rather than the one that you are pointing at. You just put it back and forth on the specimen and to cover all the surface area, and then the tunneling electricity kind of shows that, and it is connected to the monitor and the electricity is captured. Let me read it, I think I can explain it. It's extremely sensitive to the distance and will decrease about 1,000 folds if the probe is moved away from the surface by distance equivalent to the diameter of an atom. Now, the arrangement of atoms on the specimen space is determined by moving the probe tip back and forth over the surface, and the up and down of the tip is done because, like it is said that it is sensitive to the distance, so it moves up and down to the area where there is a dent and when there is a mountain type of rising area. So the tip does move up and down, but only in a restricted area because it doesn't want to go beyond that distance. It will ruin everything. And the computer analyzed that image, and the three-dimensional image of that is formed. In 1986, it was developed by Gerd Binning and Heinrich Rocher. Now, the atomic force microscope, unlike the scanning tunneling microscope, the atomic force microscope doesn't use electricity and it uses the laser. While it goes to the tip between the probe tip and the surface, all the process is the same, but when it moves up and down and the light that is scattered here and there, right, it doesn't necessarily have to be the light, it is laser. So the light that is scattered, it is collected from the photodiode, and that is used to generate an image. So the atomic force it puts a pressure and that manages the distance. In that area it was the electrical sensitivity. This one is the pressure sensitivity. So this is used for the objects that are not good with conducting electricity, like the surfaces that are not good conductor of electricity. How are you going to study the atomic level of those kind of solids or liquids? So in that case, the atomic pressure microscope is used. This is showing the aquaporine, membrane protein, aquaporine. And as you can see, this is a protein. Protein is way too small to be seen on even electron microscope, because as you can see, this is showing the atomic levels of the bonding of the aquaprotein. And this is just the surface area. And this is the even more detailed and a higher magnification picture. A single aquaporine, while these are a collective picture. Right, they are saying that it can be used to study the interaction between E. coli, Borrelia, and Goyle chaperone proteins. Like, you can only study the static proteins, you can study the proteins that can move and don't move, and you can see the interaction. So this is the end, and these were the two techniques.